as discussed in this section, we are going to talk about the tools of recombinant DNA technology. While we were discussing about the steps of recombinant DNA technology or genetic engineering, we have used certain terms like vectors, restriction enzymes, DNA ligases. So all of these are certain important tools which are required to carry out the process of our DNA technology. So let us see each and every one in detail, and first we are going to start with restriction enzymes. So basically, what are the tools? Let us know that they are restriction enzymes, polymerase enzymes, ligases, and vectors. So these were the names that we've also seen, and we'll start with restriction enzymes. Now, restriction enzymes were discovered in the year 1963 when there was something called as the restriction phenomena that was going in the E. coli. So let us understand first what is this restriction phenomena. So here is my bacteria. Let me call this as my E. coli bacteria. So I will call this as my E. coli bacteria. Just a second. Sorry for that. So we are going to write it as E. coli bacteria. Now this bacteria, let us suppose it is attacked by a bacteriophage. So it is attacked by a bacteriophage. Now what is a bacteriophage? Bacteriophage is basically a virus that feeds on the bacteria. So I'm going to draw that bacteriophage here like this. Okay, now this bacteriophage will contain its own genetic material. Okay, so this is the viral DNA. Right, and this is one is the bacteriophage. Now, you know that whether it is lytic cycle or lysogenic cycle, the viral DNA will come from the head of the virus into the bacterial cell. So, the viral DNA has entered into the bacterial cell. As soon as that happens, now what are these red color things? These red color things are the plasmids. Okay, and this blue looped structure is the genomic DNA of the bacteria. Right, now as soon as this viral bacteriophage is attacked and it is releasing the viral DNA, even before the viral DNA released, this bacteria, it produces two kinds of molecules. Okay, it will be producing two kinds of molecules which I am drawing here. One set of molecules will go and bind to the viral DNA. It will go and bind to the viral DNA, and as a result, what it will do after binding to the viral DNA, it is practically breaking the viral DNA into pieces. So what it did, it degraded the viral DNA. Degraded viral DNA. Okay, so that's what happened here. Now this other molecules will come onto the genomic DNA of the bacteria and it will cover it like this. It will cover it at different positions, not completely, but at different positions. Okay, now this process is called methylation. This is called as the methylation. Right, so what happens, what is happening here? The orange enzymes, they are enzymes that are nucleases. They are nucleases. That means they digest the nucleic acid. And what are they digesting here? They are digesting specifically the viral DNA. They are basically DNases. They digest whatever, wherever they see a DNA, they will digest it. They are specifically DNA digesting enzymes. So these enzymes, they are digesting or they are degrading the viral DNA, as you can see here. But why are they not degrading the genomic DNA? Because the genomic DNA is methylated. What happened to it? It is methylated. Genomic DNA is methylated, and methylated DNA is protected by the enzymes. Whatever enzymes, the DNA's enzymes that were produced, they are protected. So these pink color molecules here, we will call them as the methylation enzymes. Methylation enzymes. Okay, so bacteria releases two sets of enzymes. One is the digestive enzymes, one is the protective methylase enzymes. Clear? So it is a kind of its internal protection mechanism. So these nucleases, they are also called as restriction enzymes. They are called as restriction enzymes. Now, why are you specifically calling them restriction enzymes? Because they are restricting the viral growth. They are restricting viral growth. They are preventing lytic or lysogenic cycle to happen by degrading the viral DNA. So that's why they are called as restriction enzymes. And this setup that is seen, it is called as the RM setup. This is called as the RM setup, which is the restriction. R is for restriction and M is for modification. So the bacteria is restricting the viral DNA's growth and it is modifying its own genome by protecting itself from the enzymes. Understood? Now let us continue with restriction enzymes. Okay, so that is what we have spoken here, that they are responsible for restricting the growth of bacteriophages and hence they are called as restriction enzymes. Now basically these restriction enzymes, as I said, they belong to the class of nucleases. Now what are nucleases? These are enzymes acting on, they are enzymes acting on nucleic acids. So this mode of action is specifically nucleic acids. Now there are two types of restriction, basically not restriction enzymes, nucleases. Let me just remove this. So as I have changed it to the correct format. So nucleases are of two types, exonucleases and endonucleases. So endonucleases, they basically remove the nucleotides from the end, and endonucleases, they basically remove it within between. For example, let me take a DNA strand like this. This is a double-stranded DNA strand. I'm taking it on both sides. Okay, now if I'm using an exonuclease, my action of the exonucleases will be at the end of the, it will be on the end of the DNA strands. If I'm using endonucleases, the action will be in the middle, somewhere in the middle. Okay, so how will I get my fragments then? My fragments here will be small ones at the tip and one larger one in the middle here, whereas here I will have three separate fragments. Correct? So where they are removing the nucleotides? Basically nucleases, they act on DNA. How they act on DNA? They remove the nucleotides. They remove nucleotides. And how are they removing the nucleotides? That also you know it here. They remove it by breaking the phosphodiester bonds. If you remember your molecular basis of inheritance chapter, there we have discussed that nucleotides are joined together in a DNA fragment or in an RNA fragment by using phosphodiester bonds. So you break the phosphodiester bonds, you will separate the nucleotides. So that is what is happening here. So you're fragmenting a nucleic acid into pieces by breaking the phosphodiester bond, which is essentially the backbone of the nucleic acid. Now, the first restriction enzyme, endonuclease, was Hint2. Now here is the twist. Why am I calling restriction enzymes as endonucleases? That is because all restriction enzymes, they are endonucleases because they cut in the middle. They don't cut at the terminal positions. They remove nucleotides in between the DNA or the RNA or in the nucleic acid strand. So that's why we call it as the restriction enzymes are also called as restriction endonucleases. And the first one isolated was Hind2. Now, this Hind2 always cut the DNA at particular recognizing sites. Specifically, it recognizes a basis of 6 base pairs. It is not just cutting everywhere. It will not go to the onto the DNA, dance here and there, wherever it will feel like it will just cut. No, it doesn't do that. It has some specificities. Hind specifically goes and recognizes a particular area, this area that it is showing, which is made up of 6 base pairs. Let us count it: 1 2 3 4 5 6, right? So this is one base pair, right? So it is basically recognizing this 6 base pairs region and it is making the cut there, right? So that 6 base pair region is called as the recognition site. Now, it is not only Hind, but every restriction enzyme or every restriction endonuclease, it cuts the nucleic acid at its own specific recognition site. Every enzyme has its own recognition site and it will make the cut there. Okay, nobody will make the cut in others enzyme. It's like your specific marked territories. You will sit here means you will sit here. Like suppose in your classroom, everybody, if you are given a specific bench and your name is written on that bench, how will you will go recognize that bench? You see, oh, I'm supposed to sit here, you'll sit here. The same is the case for the restriction enzymes. On the DNA, you will have specific recognition sites. Each recognition site is specific for an enzyme, literally like a bench with a name written on it. So that enzyme will go and cut there only. It will not go to other people or another enzyme's recognition site. Okay. So once it finds that specific recognition site, what it does? It binds to the strands of the DNA and it breaks the sugar phosphate backbones. How is it breaking the sugar phosphate backbones? Again, as we have discussed, by breaking the phosphodiester bonds. You remove the phosphodiester bonds, you will remove the sugar phosphate breaking. And these restriction endonucleases ka recognition sites, they have very own language. See, it's like universal language for all recognition sites. As I said, there are 15 students in the class and each student speaks a different language. So I can't write that every 15 different languages on the bench. Let us say if there is a Tamil student, I cannot write his Tamil name. If there's a Telugu student, I can't write a Telugu his name in Telugu. It is going to be very confusing and weird. Instead, what the teachers will do, he will write all the 15 students' names on their respective benches in English, a common language. Similarly, all the recognition sequences, no matter what the enzyme it is, no matter how their differences, they have one common factor, and that common factor is they are palindromic in nature. Okay, so the palindrome in DNA sequence is is a sequence of base pairs of DNA that reads the same on the two strands when the orientation of reading is kept the same. Very confusing statement, right? Let us just simply understand it. See, this is a recognition sequence. Now, if I'm reading it in 5' to 3' direction, if I'm reading it in the same direction, that is 5' to 3' direction. So that means like this and like this. What will be the sequence? You read it with me. GAATTC, right? So it is GAATTC. That is the sequence. Now again below, this is upper strand, this is strand 1, this is strand 2. So on the second strand, again in 5' to 3', so in this direction, GAATTC, GAATTC, right? If you look at it, there are two different sequences. But if you are reading in one common orientation, both, whether it is upper strand or lower strand, they are reading the same. Let us now change it if it is in 3' to 5' direction. We will see if it is the same. So 3' to 5' means above it will be like this in this direction. So let us read it. CTTAAG. CTTAAG. Right. So again 3 prime to 5 prime in this direction, CTTAAG. Right. So they are reading the same on the two strands when orientation of reading is kept the same. If your orientation of reading for both the strands is 5 prime to 3 prime, they are reading the same. If your orientation of reading is 3 prime to 5 prime, then also it is kept the same. Right. Like the word Malayalam. Right. Now you read it like this or like this. It is the same. This kind of sequences are called palindrome. Now this is for a double-stranded sequence. If the sequence is a single-stranded, if the DNA molecule is a single-stranded, then the palindrome will be like this. Sorry, it will be like this. Right? So GGAACCGG. If I read it like this or like this, it is the same. Right, so this is the case of single-stranded DNA and this is the case of double-stranded DNA, how palindromes are present. And all the restriction sites, all the recognition sites of the restriction enzymes, they are arranged in this palindromic order. Right, so now here there is one restriction enzyme and this is its recognition site. Now the enzyme here is EcoR1, so it will make a cut. Now these are the fragments produced. Okay. Now if you see these fragments, they are having overhangs, means see this region here and this region here. They don't have any base pairs below. So the fragments, they are not cut equally, they are cut unequally. So that means how must have the cut come? The cut must have come like this. It must have come like this. Correct? So the cutting is not equal but unequal. Okay, so these type of ends are called as sticky ends or cohesive ends, which are overhanging. Okay, so commonly most restriction enzymes, they cut the two strands of DNA at different locations, and this type of cut is called as a staggered cut. And when they are having a staggered cut, you are having these kind of ends, overhangs. These are called as overhangs. And this ends, they are called as sticky ends. Similarly, you will have other type of cuts also. They are called as blunt ends or flush cuts. Flush cuts means what? Let me say I have my DNA like this. Right, and if the cuts are happening at the same location, they are cut equally at not at different locations but at the same location. Now what will I have here? I will have two fragments like this, equally cut fragments. Right, so these fragments, they don't have overhangs, so this cut is called as a blunt cut. Okay, and these fragments are called as blunt end fragments. Okay, so this type of cut is also seen, which is blunt end style of cut, and you have the staggered cutting or the cohesive end cutting or the stick end type of cutting. So restriction enzymes, they cut in two ways, either unequally producing the staggered cut, or that is at different locations, equally at the same location on both the strands producing the blunt cut. Okay, now why does it prefer mostly staggered cuts and why not blunt cuts? Well, the Staggered cuts, they are very easily joined by using DNA ligases, because if you see the overhangs like this, they are actually complementary to each other. So like they are two perfect pieces of puzzle. So when you join them, it is easy to form the hydrogen bonds between them, and DNA ligase will come and seal it very easily. Right, like two perfect pieces. See the animation, they are just two perfect pieces which are complementary to each other. So when they join together, they very quickly form the hydrogen bonds between the base pairs, and ligase will seal the ends. That will not be the case of blunt ends, because blunt ends are like two different ends. So if you want to seal it, first you need to bring them together and join them using a separate molecule which is called as an adapter molecule, and then the ligase will act on it. So many different things are happening in the blunt end cuts, but but the staggered cuts or the sticky ends, they're so easy to join. So that's why the restriction enzymes, they prefer mostly the staggered cuts. Now, even though the first enzyme was isolated as Hint2, today you have 900, more than 900 restriction enzymes that have been isolated over 230 different strains of bacteria. Few examples are given here. See, BAM-H1, ECO-R1, HIND3, PST1, SMA1 and HPA1. And if you see the first three, one, sorry, two, three, four, all of them are cutting in unequal direction. They are cutting at two different locations. So what do? All of these will produce the staggered cuts. They are maintaining the staggered cuts. But if you look at these two, the last two, they are cutting at the same direction, same level. So these two will produce the blunt end cuts. And if you see, each one is having a different recognition site. Okay, now can you calculate theoretically how frequent the ECO-R1 recognition site is in DNA? Can you do it? Can you try? What I'm going to do is I'm going to stop the video for a few minutes so that you people do it on your own. And I would also suggest you to do that. You stop the video, look at your textbook. It has the recognition sequence of ECOR1. Try calculating it. How many times can this be coming? That is the hint I'm giving. Well, I hope you have done it. If not, let us see how it is done. Now, as we have seen, the hint I have given is there are four types of nucleic acids. What are those four types? Let us write it here. It is A, T, G and C. Okay. Now, the probability of any of these occurring at a place in 1/4 means, let us say I have this as my this is my sugar phosphate strand. Now here there is the chance for 1. Okay, I'll just take a single one. Wait, let me erase this double strand. I'll put a single strand so that it is easier for you to understand. Yeah, so here only one nucleotide can come. So this is the place for the nitrogen base. Now which one can come? Either A can come, or D can be there, or G can be there, or C can be there. So the chance of having any of them is 1/4 probability. Correct? Now, assuming that the occurrence of the bases in adjacent positions is the random, the probability that you get G double A double T double C. Now, what is this G double A double T double C? It is the recognition site for the ECOR1. So, assuming the probability of having one base, the probability of one nitrogen base is 1/4. The probability of having 6 nitrogen bases, that too in random, to attain this particular number, this particular order, it will be what? 1/4 to the power of 6. Correct? Right, it is 6 times 1/4. 1/4 probability of G, 1/4 probability of A, again 1/4 probability of A, 1/4, 1/4, 1/4. So you are having 6 1/4s, which is equal to 4096. So for every 4096 nucleotides, there is a chance that you will get one recognition site for EcoR1. Interesting, right? This question is given in your NCERT textbook. Now, before we go into the MCQs, I wanted to discuss another part, that is naming the restriction endonucleases. How do they name them? There is this EcoR1, HIND, PST, SMA, something you have seen. So how are these different names coming from? Now, before you understand the names, you also have to understand these Roman numbers. What are these Roman numbers? So let us see those two, and then we'll go to the MCQs. Now, it is said that restriction enzymes are present in or categorized into three different classes. Restriction enzymes, they are categorized into three different types. Right? So they are type 1, type 2 and type 3. So the Roman number which you will see in the last of at the end of every restriction enzyme resembles its type. So what is the difference? Let us see each one. Now type 1 restriction enzymes, they basically they cleave only one strand of DNA. So they act upon single stranded DNA. They act on single stranded or one strand of DNA. They don't act upon both the strands. Okay. Now type 2, it cuts the DNA on both strands. Cuts DNA on both strands, unlike type 1. Okay, so whether it is 5' to 3' direction or 3' to 5' direction, it cuts the DNA on both strands. Now type 3 is similar to type 2. It is same as type 2. The only difference is type 3, it needs ATP for action. So to carry out the restriction, that is the restriction digestion, to cut the DNA, it needs ATP. So based on this nature, restriction enzymes are divided into type 1, type 2 and type 3. So now let us name the restriction enzyme. Now let us say I have taken a restriction enzyme from Escherichia coli. Escherichia coli bacteria, strain RY, and this belongs to type 1. Okay, so how are you going to name it? First thing is the first letter. The first letter of the name will be the first letter of the genus's name. First letter of the genus name. Now what is the first letter of the genus name? E. So it is E. Let us write it here. E. Next, the second and third letters. The second and third letters, they are the first and second letters of the species name. Of the species name. What is that here? CO. We'll write it here. CO. Then the last letter, fourth letter, will be the name of the strain type. First letter of the strain type. Now here the strain is RY. So the letter will be R. Okay. And lastly, coming to the Roman number, mention the type. Mention the type. What is the type here? 1. So the enzyme here is EcoR1. Okay, so let us take another example and do it. Let us take another example. This time we will write it as, okay, It will be, okay, let us write it. Haemophilus influenzae strain D type 2. Okay, now I'm going to give you 2 minutes. Pause the video and answer this. I hope all of you have done the answering. So it is HIND2. So it will be, let us write it here, HIND2. Okay, let us take another example. This time we will take an example of, okay, let me find one example for you. Okay, it is Serratia maricensis. Interesting. Okay, Serratia maricensis. Okay, I have to look at the spelling. Okay, there is no strain, but we are giving the type. Now what happens here? Because there is no strain here, you're going to just take SMA, because there is no strain, the fourth letter will be empty, and you'll just take the type 1. Okay, another one. I'm finding this very interesting, right? Even I hope you're also feeling interested. So let us write another one. It is Haemophilus parainfluenzae type 2. Okay, so what will this be now? Come on. So this will be HPA, right? There is no strain, so fourth letter will be empty, and type 2. Got it? So this is where your restriction enzymes are going to be named. Okay, I hope this is clear. Now let us see some MCQs in this regard. So the first MCQ is, genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools like restriction enzymes, polymerases, ligases, vectors and the host, all of the above. What should be the correct answer? Without each of them, that won't work. So the correct answer is all of the above. Next question. In the nomenclature of restriction enzymes, the first letter of the name comes from species, strain, family, genus. Here family means the type 1, type 2, type 3. So the correct answer is genus. Right. Exonucleases cut the DNA at the middle of the DNA, from the ends, center to the end, all of the above. Correct answer is from the ends of the DNA. Right. So next we are going to talk about in another video cloning vectors.
For more content, like, share and subscribe to www.i-win.in.